It's a pleasure to introduce to you our new product, Jellagel, which over the course of the next half an hour, I'll tell you about what this material is in terms of its hydrogel properties from jellyfish, as we believe it to be a new alternative for 3D cell culture and regenerative medicine. As an introduction to Jellagel, for those of you that don't know the company, we're a UK-based marine biotechnology company, and we manufacture to ISO 13485 certification a collagen derived from jellyfish. As you see, I founded the business over 5 years ago now, and we have a manufacturing facility in Cardiff, which is where we're located, and then employ up to 12 people in our bespoke facilities, which we manufacture to that standard. And one of the reasons that I want to start setting the scene is that we hear from multiple sources that collagen is still the best biomaterial to grow cells for today and tomorrow. And why is that evidence? Well, it's clearly evidenced in some of the recent literature where its collagen's high biocompatibility makes it a good material to implant into the human body. It also promotes good cell adhesion and survival because it's biocompatible, because we are cells composed of collagen. And it enables a range of cell culture methodologies to be handled in the lab from 2D to 3D. And we're seeing a greater turn of material scientists turning back to collagen again, if based on the literature reviews. So we thought jellyfish would be a very good alternative to get over some of the reasons why collagen is sometimes being turned away from. So it is the collagen to grow cells. It makes a third up of the protein in the human body. It's found in bone, muscles, skin, and tendons. It can be chemically conserved, and of course is non-cytotoxic and bioresorbable. And historically, although it's been used for a range of functions, it's historically originated from humans, cows, and pigs. With the challenges around using collagen, especially from rat tail at laboratory levels, with mammalian sources, you have the viral and disease transfer risks. We hear from the customer base that we get batch-to-batch inconsistencies from the use of rat tail and also with other 3D and also with other materials such as matrigel. Rat tail is also non-translatable technology if you're working on regenerative medicine applications. And we understand that the FDA and EPA are looking and scrutinizing more on mammalian-derived systems. With the advent of synthetics, which are often based around beta-structured fibrous materials, that has issues around biocompatibility, and of course, recombinant collagens are very expensive. So with all that in mind, we have a very complex physiology where we get collagen from mammalian sources, which does lead to variability within species, to jellyfish, which is a low evolutionary system. It's predominantly produced from collagen, and unlike its mammalian counterparts, is considered to be planktonic. And because of its ancient chemical lineage, the collagen itself does provide some innovative benefits, which I'll talk to you about now. First of all, collagen from jellyfish is collagen and is very much applicable in cell culture and biomedical applications. And we see this through a growing body of evidence in the public domain. We ourselves are about to publish our own data shortly, but this just adds to the growing list of reasons why jellyfish collagen does present a useful alternative with benefits over existing materials. It's a growing list, and again, very recent, all the way from cell culture and through isolation and characterization of the material itself. But why, I've touched on this, but just to reiterate again, a jellyfish, unlike a cow, has a low level complexity and is chemically ancient. So you've got a jellyfish, which has a very simplistic lifestyle, producing multiple offspring, and the jellyfish being quite physiologically simple, the protein diversity within a jellyfish is predominantly by dry weight collagen. So this translates into a much more consistent batch-to-batch product, consistent product, which gets some of the over some of the disadvantages with alternative mammalian sources. We also, there are a number of other benefits associated with jellyfish collagen, one of which is that it's clean and inert in terms of how cells respond. And we see this as well at the microRNA level, where we compared it to a bovine system, where we see a much cleaner profile for microRNA. And those that you don't know, microRNA are small oligonucleotides that do affect cell phenotype, but with our materials, there are less of them in it. And could jellyfish collagen become the next gold standard? Well, we believe so. And the reasons to adopt jellyfish collagen are very much simply can be categorized as follows. It's prion and disease vector free. It is inert and significantly cleaner at the microRNA level, which you've just seen the data exemplifying that. With the data that we're generating through ourselves and through our customer base, we're demonstrating that it does offer biocompatibility, offering low immunogenicity, non-pro-inflammatory effects. And it's also non-cytotoxic as well. Because of the simple physiology of jellyfish I mentioned, that does translate into consistency from one batch to another. And on the characterization of the material itself, based on amino acid sequencing, on an LC-MS, MS profiling, that the sequences that we get from our collagen from jellyfish does offer sequence homology to human type 1, 2, 3, and 5. And we think that is predominantly down to the ancient chemical lineage of that starting point. Our material is compatible, and importantly, with all existing cell culture methodologies. So that's good news, and is ethically sourced. And through our own activities and through the activities of our customer base, we're seeing a growing body of evidence of a number of human cell lines growing very well on our material, ranging from nerve cells, microglial, central nervous system, immunocells, skin, bone, tendon, and the range continues. So given all that, the focus of today's webinar is obviously to launch Jellagel, which is a format of our material that we've worked on for the past 2 years to provide a simple, usable, user-friendly material to create 3D hydrogel matrices. So in terms of the material itself, it has a number of, we think, very good features that differ away from existing hydrogel sources. First of all, it's batch-to-batch consistent for the reasons I've just explained. It's from a non-mammalian source, translatable, and has an inertness and ease of use, which, well, the ease of use is a very particularly important one, where unlike mouse sarcoma-derived products, you don't have to be in a cold room to use it. You can be at room, you take the material straight out of the fridge, and that's it. You don't need to work in a cold environment. You can work at room temperature, and that is better for the scientist and better for the cells. They're less stressed. So in terms of the ease of use, handling at room temperature, again, it's a very simple protocol where you take our material, neutralize it in a buffer at room temperature, follow a very simple first phase 5-step process to neutralize. And then the second phase, again, is incubation, ensuring you reach a pH of 7.4, incubate for 15 minutes with your cells encapsulated in the material, and out comes the gel ready formed with your cells encapsulated within. And this is a very interesting feature of the material, this self-encapsulation of the cells in all 3 dimensions, which is synchronous. I shall explain that in more detail. So traditional hydrogels work where you create a gel at the bottom of the well or the vessel that you're using to create the gel, which creates a very 2D differential between waste and nutrient exchange. With Jellagel, because it self-contracts into this and shrink wraps the cells and arrests the cells at that time point, you get a much more even distribution of cell growth factors, nutrients, and aeration and gaseous exchange in the material. And this works in a very different way from all traditional gels. And in terms of rheological properties, the stiffness, again, we're straight in with the Matrigel kind of space. We're offering, it has good elasticity, good pressure matrices in terms of its characteristics, and how that looks when you grow cells is that, first of all, this is just an example, but the first thing we see is that when you culture cells in Jellagel, the cells are encapsulated without affecting their viability. This provides a very good biocompatible microenvironment. And interestingly, the cells retain their activity that they exhibit at the time point they're encapsulated. And we see this with the expression of cellular filopodia early on in the cell culture timeline. And as you know, filopodia are the thin acting rich plasma membrane protrusions that act for cells to sense their environment around them. And that's a very good sign that the material provides the environment required for good cell growth. In terms of locking the cells in 3D, this video just shows you very clearly that you see a very good three-dimensional encapsulation of the cells, and they are viable. And this is just an example of HeLa osteosarcoma cells at 2-day time point. But when we look at an example, as we looked at HeLa cancer cell lines, after 2 days, we saw very good growth throughout the gel matrix. The blue dots are the nuclei and the green are the actin stain, the cytoskeletal architecture of the 3D matrix. And the gray background is the Jellagel in the background. So we looked at this in more detail. We looked at a time course, and we could see differential morphology from cells growing on the surface of Jellagel to those growing inside Jellagel, which is also very interesting. So at day 1, you already start seeing the cellular filopodia being produced. And we then left that to day 4, where you see some proliferation of those cells. And by day 8, you see that the cells are proliferated very well in the gel and are producing all the cytoskeletal architecture one would expect from that cell culture. So this just shows you the imaging throughout the gel, which shows you very nice sort of spread of the cells throughout the gel in its entirety, and also exemplifies that imaging of cells cultured in Jellagel is very straightforward using standard imaging techniques. We then looked at, okay, so are these cells proliferative? And we answered, and that was yes in the affirmative. They are proliferative inside the gel, which is also very good news and very encouraging. We then looked at another cell line, osteosarcoma cells, cultured over 3 days, cultured in 3D over 1 to 8 days, and again saw exactly the same kind of growth pattern: cellular filopodia being produced, cytoskeletal architecture, to well proliferated cell structures in the hydrogel after time point day 8. So this goes to show in 2 cell lines, HeLa and osteosarcoma, some very good results. And this again just shows you that the osteosarcoma cells could be seen throughout the gel in wide field Z-stack imaging. So that's cancer cells. So we also looked at human primary cells as well, and we looked at, well, what would human skin and bone demonstrate? And again, we successfully encapsulated in 3 dimensions human primary fibroblasts and osteoblasts, and these were calcine AM live staining of the cell types. And over 8 days, we looked at imaging at the surface and getting deeper into the hydrogel. And as you can see, that both the skin and the bone structures were starting to differentiate into morphologically correct types the deeper you got into the gel. So they were feeling their environment and responding appropriately. And again, we're very pleased. Actually, no, we demonstrated that exemplar there. So in conclusion, Jellagel, the company, and the raw biomaterial, the collagen itself, has some very unique characteristics. It's biocompatible against multiple cell lines. It has a consistency advantage and is manufactured according to ISO 13485, which, just to explain, does contain elements of cGMP, but is covered under ISO. The material is non-pro-inflammatory and is clean and inert. Jellagel, the product that I've just explained and demonstrated to you, provides a very interesting chemically defined alternative to existing products on the market which have an undetermined chemical variability. Jellagel also provides a very good biocompatible microenvironment and crucially can be worked on at room temperature as opposed to working in a cold room. The material at the chemical level is also tunable. You can dial up the stiffness if you so wish, and that is something that Jellagel's team can help and support on if that was of interest. So our material, again, is collagen, and for the reasons I explained at the start of this webinar, collagen is the best material to grow cells on. This is collagen. It's just derived from a very interesting marine source that has a evolutionary advantage over existing supplies, and is again an evolution, not necessarily a revolution in that regard. In terms of sales contacts, Clini Sciences are distributors for material, and I thank you for your attention for this webinar, and any questions we would be delighted to answer.
If you have any question, you can ask by your sound, or you can send a message by the chat.
Does anyone want to answer some questions? Ask some questions?
I have a question concerning the application for the Jellagel. Is it possible to use paraffin in order to make some sections of the culture of the cells?
It is. We have done that in the past for chondrogenic progenitor cells, which I haven't presented here. But yes, it is possible. We have a protocol for that in-house.
Okay, thanks. I have a question on the chat. Have you tried to embed more than one type of cell?
In co-culture, not in the hydrogel yet, but we have for the Jellagel biomaterial in 2 dimensions. So this is where we want to. This is exactly where we want to go to. We have successfully co-cultured astrocytes and neurons on our material, and we're getting some very interesting results on that basis, and we would like to move that into this 3D hydrogel. So the answer is yes, we are working towards that.
Can you share what type of cells or the
area
of research?
Yeah, so we're very interested in the sort of brain health, brain area, and we're in the. I'm not presenting it here because we're still in the middle of getting the data, but we've successfully cultured astrocytes on Jellagel material, and one of the interesting things was that cortical neurons were not toxic to our material, which is good. But one of the advantages that we're seeing is that when we do co-culture, we know that the astrocytes are producing the ECM matrix components for the enabling of the cortical neurons to attach. So it gives a precision advantage and less variability of when we get to that next stage in 3D, which I'm quite excited about. So yeah, more to come on that.
Another question, have you used it for skin reconstruction?
Okay, so we have a number of interesting collaborations going on right now looking at skin. And I guess the answer to that is yes. At this stage. So any further, if you'd like to know more on that, if they could come to us directly or through Clini Science on that directly, that would be good, because obviously there's a lot more to say around that subject.
Okay, thanks. And I have another one. Are there any experiences in cultivation of primary hepatocytes in a sandwich culture? And if there is a better yield of mRNA when using Jellagel instead of normal collagen?
So the answer is that we have done that experiment. We compared it to a Geltrex sandwich cell culture protocol, and we used it in primary hepatocytes. And over a 5-day period, we got confluence equaling that of a rat tail Geltrex sandwich model. When we looked at cytochrome P450 output, there was no difference between our material as well, but we didn't get to look at the messenger RNA level at that stage. But what we're seeing is that it is possible to do with our material, yes.
Okay, thanks. I have no other question on the chat. Do you have any question? You can use the chat or you can ask directly. There is no other questions, so I think that we can end. Thanks a lot to everyone to join this webinar. We record this webinar and the video will be available on our website. If you have any other question, you can contact us to the different address here, depending on your country. Thanks a lot.
Thank you very much.
Thank you.
Have a nice day. Bye-bye.
Thank you very much for everyone. Thank you.